All right, so we want to thank everyone in the room and those are online for joining us for our eighth episode of spring semester CHEMSEM chemistry seminar. Today we have the distinct privilege of having as our guest speaker Amira, Farzana, Samat. And guys, it's 5:30 or so right now in Malaysia, where she's up to give us a lecture here in the morning. Yeah, so we need to even give her a round of applause just for that. Thank you so very much for getting up this early to share your work with us. So as you could see from her slide, she would be talking about biodegradation of polypropylene. She is awaiting word from her examiners on, she is a PhD candidate and she submitted all of her work, and hopefully very soon we could start calling you Dr. Samat. Before she went to do her PhD, she was a lecturer at University Malaysia, Perlis. And I think I saw some maps, so she'll show us where those are. She was pursuing her PhD in Sydney, Australia, the University of Sydney. Her background, academic background, is in biotechnology and majoring in environmental biotechnology. Her current interest, and I guess what we'll be listening to today, deals with the utilization of microorganisms for biodegradation of polymers, which is a very important topic. And she will explain the significance of it. And she hopes that her work will help to mitigate plastic waste problems by introducing a more environmentally sustainable approach. So we will call you Doctor. Dr. Samat, thank you, and the time is yours.
Right. Thank you very much, and good afternoon to everyone there. Okay, first, of course, I would like to thank Dr. Desmond Murray for inviting me to be part of the guest lecture for your program. I'm truly honored to be invited, really, because this is the first time I've been invited for a foreign guest lecture. So thank you so much. So yeah, today, yeah, I cannot see everyone there, but I'm sure that you guys are like a bright young minds who actually, probably are more way smarter than me. I'm sure of that. Okay, so so today, yes, like Dr. Desmond mentioned before, I would like to present to you about my PhD journey, the topic on the biodegradation of polypropylene. So, okay. So Dr. Desmond mentioned that I'm from University Malaysia, Perlis. So this is Perlis. If you can see like the tiny blue area here, at the very northern part of Malaysia, we are actually in border with Thailand. So I can get to Thailand in about an hour every day. So yeah, this is my university. If you've ever been to my university in Perlis, so this is how the entrance look like. Okay. But actually my home is in a small town called Jitra. It's actually in Kedah, in the pink area here. So Kedah is in another state. So I travel for about 40 minutes every day to reach the university. But it doesn't feel far anymore because you've done that every day. So it was fine. Well, anyway, enough of the geography lesson. So I would like to introduce a little bit about my research team. So these are the team, like everyone in it. So Ali here, Ali Abbas is my main supervisor in the University of Sydney. So my co-supervisors are Dee Carter, who is an expert in mycology or in fungi. So Gustavo helps me with the environmental part, and Alejandro helped me in terms of chemistry part. So everyone here are based in the University of Sydney. It is a very beautiful university. It looks like Hogwarts. So if you want to try to visit them, it will be great. Right. So I will just explain a little bit about some of the introduction, the research summaries, the findings, and a little bit about conclusions. Okay, but before we start, I would like you to have a look about these videos. So this video was prepared by Matthew Agius from Cosmos, okay, from the Royal Institute of Australia. So he made this video after my research has been featured in ABC News Australia. So this video will give you a little bit of idea about what this research is all about.
Plastic is a massive problem. Every year, more than 400 million tonnes of plastic is produced, and most is single-use. Only about 10% is recycled. There are plenty of solutions that have been thrown around. The most obvious is to stop making so much, or to make it multi-use. Others have suggested accountability structures where companies are made responsible for their plastic waste. But could science have another solution? In Sydney, researchers are investigating the use of fungi as a plastic eating solution. This is Professor Ali Abbas. He's a professor of chemical engineering at the University of Sydney.
Research in the degradation of plastics is very important because it sheds light on how biology can help us to biodeteriorate these plastics. And that's important because biology works at low temperatures, as we know. When we have recycling that occurs commonly at very high temperatures, that expends a lot of energy. So we do want to create a solution that's low temperature and natural type of process.
His student, Amira Farzana Samat, has recently led an investigation into 2 fungi species and their plastic-eating abilities. One of those fungi is called Aspergillus terreus, a soil mold. The other is Engiodontium album. Both have been found to break down polypropylene, a cheap plastic commonly used in manufacturing. Amira brought us inside her lab to see how they prepare their fungi for a most unusual type of meal.
This is basically the biosafety cabinet where we are doing most of the fungal or experimental work here. We incubate these fungi with the plastics. We kind of want to see what is the degradation rate that we are looking for. And after we put it in the flask, we will incubate the plastic in this incubator shaker. We actually set the temperatures, the RPM, and how many hours or days we want to incubate this fungi. So basically in this flask, there's plastics, fungi, and the media that's supposed to support the growth, but we eliminate the elements of carbon because we want the fungi to fit in the carbon from our plastics. This is one of the fungi that we use to degrade polypropylene. So this fungi is called Engiodontium album. This fungi can be found mostly in plants. We also have another types of fungi that we use. This is called Aspergillus terreus. So this fungi is typically found in soil. So these two can be easily found in the environment. These are not natives to Australia. So we are currently looking to utilize these two to degrade polypropylene, and we are looking to improve and optimize the process so that the degradation rate can be increased from what we already have now.
When exposed to the fungi, the plastic was found to have reduced by a fifth after 30 days and by a quarter after 3 months. But unfortunately, for those looking to fungi or other microorganisms like bacteria, the results aren't yet at the scale needed to eat through the world's plastic. What Ali and Amira hope, however, is that their work can be industrialized to have an impact on plastic pollution at scale.
The scale-up of such processes requires lots of testing in the lab first up to understand those degradation rates. And thereafter, we take that into the scale of shake flask culture, such as here, where we are growing the fungi at this scale on the plastics as a substrate. This allows us then to have more confidence on the degradation rates at this scale, and thereafter we can take it to another scale in terms of scaling up as pilot facilities before we can commercialize this process. And that's where the big success will be in a few years' time, where we can say that this process is scalable and it can serve the industrial level.
Right, so that's basically about it. I hope you enjoyed the video. Okay, so yeah, that's my supervisor. So we've been like been side by side in doing the videos, and we want like the public to actually know what we are doing in a simple term, instead of like just reading the published manuscript, because not everyone reads that, I'm sure. All right, so so basically, as you can see from the video, this research is about polypropylene. It revolves around polypropylene because it is one of the top plastic waste producers globally. So of course, you can see that this plastic mostly end up in landfill and, not surprisingly, marine or river water. So we are thinking about closing the loop. So it would be ideal to use something that have low energy consumption, exists in the environment, and produce non-toxic byproducts, like Ali mentioned before. So, and then of course, we end up using microbes. So in my research, polypropylene with different properties were tested. As you can see, where the one that we're holding like granulated form, film, and also metallized film form. Okay, so we then pre-treated these plastics using 3 types of pre-treatments. We have UV, heat, and also Fenton's reagent. So we chose these 3 types of pre-treatment to pre-treat our plastic before we feed them to the fungi or microorganisms. So as Ali mentioned before, we use 2 types of fungi, the Aspergillus and Egerontium album at first, and then we also isolate new microorganisms, and we utilize them the same way. Okay, and then we incubate them at a specific time duration. We have like 30, 60, and 90 days, and then we proceed to test. We did a series of tests to see any signs of degradation. So ideally, we want this partially degraded polypropylene to be further utilized downstream. So I can say that in general, we can see some changes in terms of the physical appearances and the chemical structure of polypropylene. So we know that it works. It's just it is happening in a small scale, in a lab scale. Okay, so well, I can summarize my research that we relate this kind of research with current global situation of plastic waste. And we finally decided to delve into the use of untested fungi, like I've mentioned before, and the newly isolated microbes. And we want to predict the degradation mechanism, like what happened inside, what happened to the polymer and what happened inside the microorganisms. And of course, I've also included some future works that could be done to further explore its plastic degrading possibilities. So since my work actually started during COVID, specifically in March 2020, So there are some experiments that have to be removed due to time constraint, such as this one, the genetically modifying fungi. So because we know that there are some enzymes that are produced by this fungi that are able to degrade the polymer. So if we are able to genetically modify this fungi or the bacteria, we can actually enhance the production of enzyme, and we can see an enhance in terms of the polymer degradation. So hopefully someone could continue doing that soon. Right. So so this is how I designed my research. So I divided the research into 4 parts based on the objectives and hypotheses that I want to answer. Okay, so basically the first part I want to explain the degradation of polypropylene using only Aspergillostereus and Angiodontium album. So this one is like a preliminary testing. And then for the second part, the light blue one here, we then assess the degradability by using FTIR, TGA, SEM, and GCMS. So I will explain this in detail later. And then for the third part, we are looking to isolate the microorganism, and we also test them for their biodegradability, whether they are able to actually degrade polypropylene, and then we compare them with the previous fungi to see which one is better. And then last but not least, we did a lacase enzyme assay to see which fungus actually produce the highest amount of lacase that we believe to have contributed greatly in the degradation of this polymer. Right. So, so now we go into a more technical part of the research. But I must warn you that I think this lecture is more biological oriented, because rather than chemistry or chemical engineering, as we like to actually emphasize on the aspect of the ability of this microbe to degrade polymer. So please bear with me. Right. So so basically this part has mostly been described in the published paper. So if you want to have an in-depth read on this part of my research, I can send you the link. I can give it through Dr. Desmond if you want one. Okay. So basically, first I did a preliminary assessment of the pre-treated polypropylene. So this assessment comprises of biomass production, one of this one. And then we also did a weight loss analysis where we weighed the polymer before and after to see the differences. And then we also conducted a reduction rate, half-life, and the correlation analysis. So this will comprises everything in this first part. So as you can see here, this is the fungal biomass production. So this graph represents the biomass produced from both Aspergillus and Angiodontium album. So overall, you can see that the highest biomass was produced from Aspergillostereus when they are incubated with the heat-treated polypropylene. So if you could recall, I utilized 3 types of polypropylene: the film, granule, and metallized film. So the highest are shown to be exhibited by the metallized film that are heat-treated. Okay. And you could also see the overall trend on your right side here. When we compare to the control, the heat and UV treated polypropylene, regardless of its form, like film, metallized film, or even the granulated form, are shown to cause the fungi to produce higher biomass compared to the control. So this first indicator is actually very important, because we know that the higher the biomass produced, the more likely the fungi can grow and consume the polymers. So we want the fungi to be able to grow throughout 30, 60, and 90 days. So we indicated the growth based on the fungal biomass. So when we weigh on the biomass, there are increment in terms of 30, 60, and 90 days. So we know that some of the fungi are able to grow and fed on the polymer that we feed them. Okay, so in other words, the higher mass increase after 90 days in the presence of polypropylene could be attributed to the use of the polypropylene itself as the nutrient source. So they can feed on this fungi, so they can feed on this polypropylene. Right. So the next one, we also did a gravimetric weight loss. So this is basically just weighing the polymer before and after, and then we see the differences, like how much of this polymer has been consumed or has been degraded by the fungi. Okay, so so same goes with the fungal biomass data that we saw earlier. We see that highest weight loss was also produced by Aspergillus terreus compared to Angiodentum album. And the same thing, the trend shows that the highest weight loss of polypropylene are incubated with both UV and heat-treated metallized film. So these two has been shown to cause the most weight loss, and we could concluded that these fungi are able to feed on this metallized film compared to film and also granulated film. Right. And then we also measure the reduction rate. So the reduction rate is actually measures how much polypropylene has been degraded per day, while half-life determines how long does it take to fully degrade half of polypropylene samples. So you can see the formula for reduction rate here. So it is divided by time. So we calculate for every 30, 60, and 90 days. So we can see that the highest reduction rates are observed for the metallized film after 30 days. Because you see that although the weight loss increase from 30, 60, and 90 days, the highest reduction rate, of course, will occupy by the 30 days because we divide it by time, which is the days, right? So then the half-life will show the opposite of the reduction rate. So you can see that the half-life, of course, the highest will be the control, because this is almost infinity actually. None of the polymer were degraded for the controls, but it is highly degraded, or the half-life is much, much lower for those polymer that are pre-treated by using heat and also UV. And then we also did a correlation analysis to determine what are the best pretreatments that correspond to which polypropylene samples that is affected the most during the degradations. So here we can see that a metallized film will generally produces highest biomass and indicated highest weight loss compared to other polypropylene samples. So you can see the highest, the green one here, the metallized film, and then they are heat treated and UV treated mostly. So those produces the highest biomass and highest weight loss. So those are that we want to see the most. Right. And then we move on to the part 2 of my research, where we want to see the chemical and morphological characterization of pre-treated polypropylene. So these are the results for only Aspergillus and Angiodentum album first. So the first one for the analysis I did on the FTIR. So I'm not sure if you are well versed on the equipment, but this FTIR, or Fourier transform infrared spectroscopy, is used to describe the changes in the structure of the polypropylene samples. Okay, so you can see that I've emphasized on some of the peaks. So when I put this sample on the FTIR equipment, FTIR machine, there are peaks that can be observed. Okay, so in order to see whether there are degradation or not, I focus on this wave number 1167 and 1745. So basically, compared to the control, we want to see that this peak increases after the incubation with the fungi, so that we can conclude that there are carbonyl and phenolic bands observed, because we know that the structure of the polypropylene is only carbon and hydrogen. So when we see that the presence of carbon or carbonyl and phenolic bands on the surface of the polypropylene, we can be sure that there are some kind of changes in terms of its structure that has been caused by the fungi, and also, of course, contributed or helped with the pretreatment that we've done on the polymer. Okay, and we can see that the metallized film exhibited an increase in terms of the 1745, as I mentioned before. Right, and then we also conducted the thermogravimetric analysis. Okay, so this equipment actually used to describe the thermal stability of the polymer. So basically, I took the polypropylene after the incubation. I put it in this TGA analyzer, and we will heat them from from 30 to 900 degrees Celsius. So in between this process, we can see at which point, at which temperature, that this polymer starts to degrade or starts to decline in terms of the weight loss, right? So if you can see here, in the graph, I've highlighted, I've actually pointed out to some of the point in this graph. So this indicates the onset temperature. So we extrapolate this onset temperature to denote the temperature at which the weight loss begins. So for the pristine polypropylene, the onset temperature will be much higher. So for example, here, the onset temperature for the control will be like around 430 degrees Celsius. So after 90 days, after the incubation with the fungi, you can see that the onset temperature starts to decline. So it reduces the thermal stability of this polypropylene. So in a way, we know that after the incubation with the fungi, they are able to actually cause some kind of, we call it chain scission on the polymer, which are likely to occur also on other types of polyolefins, such as polyethylene, right? So we want this to happen. So a much lower onset temperature, if you can see from the reduction here, indicated the degradation of the polymer by the fungi. Right. And of course, in terms of the surface structure or the surface morphology, we also want to see what happened there, because most of the time we can't really see what happened on the polymer using our naked eyes. We can see a little bit, like a little damage done on the polymer on the surface, but by using scanning electron microscopy, we are able to actually see in depth what happened. Sorry, what damage has been done by these fungi after 30, 60, and 90 days. Okay, so first we, of course, we take the sample from the control. So the control we did on the without pretreatment. So this basically shows nothing. And then we also did the surface morphological changes on after the pretreatment process. So you can see that there are some damage done after the pretreatment. So that is what we want, because without the pretreatment, basically the fungi cannot act on the surface or can't even do anything on the polymer. So there are some damages can be observed, but after the incubation with the fungi, you can see a much more damage has been done on each of the surface of these polypropylene materials. Okay, so we can see that with the pretreatment, the damage was enhanced. Okay, and it indicates it also indicates that in some of the surface there are fungal biomass still attached to them. So it means that these two fungal species are able to attach themselves, penetrate the polymer, and use them as the carbon sources. And then we also did the EDS. So EDS is actually an energy dispersive X-ray. So this EDS actually helps to determine the element, the percentage of the element presence on the surface of the polymer. So here I want to see the atomic concentration number, especially on the carbon, to be reduced. So you can see that from the control, we have about 78.2%. But after the incubation, you can see that the carbon concentration has been declined. So this actually indicates that the fungi are able to consume the carbon sources from this polymer after certain periods of incubation due to the decline in the carbon number. Right, and then the last part of the experiment for this part 2, we also did a gas chromatography analysis. Right, so these gas chromatography are used to actually analyze the end product produced after the incubation with the fungi. So you can see that the table here actually displays the heat-treated samples of Aspergillus terreus. So I just want to highlight some of the compound that we found, especially this methyl nanodecane and also cis-9 tetradecanoic acid. So these are actually fatty acids that are found after the incubation with the fungi. So the detection of this fatty acid suggests the formation of the carbonyl groups, which are further oxidized, forming ketones and aldehydes. So if you still remember in FTIR, we could verify that there are carbonyl groups present. So here in gas chromatography, it confirmed what are the compounds that actually contributed to the carbonyl groups that we found earlier. So we can also see that there are a lot of lower carbon number from C10 to C40, if you can see the molecular formulas here. So these low carbon number are, we can say that it is a preferred carbon number for the microbial uptake to be assimilated. So a higher carbon number, like more than 40, are not suitable for the fungi to be able to consume. So if we have a lot of carbon number less than 40, it means that these microorganisms are able to uptake them, assimilate them, and use them as a carbon source and as their energy sources. Right. And then, okay, so these are a much more biological oriented part, because this part I've actually identified and did the biodegradation assessment on the polypropylene. So this chapter is quite long in my thesis actually. All right, so of course, when we did a screening, when we isolate this microorganism, we got like, I think 3 to 4 types of microbes, but we select the most dominant one. So we finally decided on 1 fungal isolate and 1 bacterial isolate. So this fungal isolate, of course, we did like a preliminary staining. We just want to see whether they are spore related or not. They have hyphae like this one or not. So we did a lactophenol cotton blue staining on the fungi, and we did a gram staining on the bacterial isolate. So for the bacteria, we can clearly see that This is a gram-positive bacteria, and the shape is a rod shape, and which are suggested to be a bacillus. Right. And then in order to identify them, so this is quite interesting for me because I have spent so many times doing this, because this is like molecular work 101. Anyway, first we did MALDI, because when I successfully isolate this microorganism, I want to find like the most simple way to actually identify the microbes, because I think everyone knows that in order to actually identify the species, we have to do DNA sequencing. Okay, everyone knows that, and that is the most reliable way. But my co-supervisor actually suggested to do MALDI-TOF using MALDI-TOF instrument. It is actually a matrix-assisted dispersion time of flight equipment. So this is randomly used to identify the microorganism. But in University of Sydney, we do have this instrument in the veterinary school. So why not utilize them? So what we found here, we got to identify the species. We have Aspergillus fisheri and Bacillus pumilus, but the score, if you can see here, is actually quite low. So this is how MALDI score the results. So if you got like 1.79, is actually low confidence, but for the bacteria, on the other hand, is high confidence, but still it is not as high as we expected. So we then decided to actually do DNA sequencing, because we want to be sure what kind of species that we are dealing with, whether it is pathogenic or not. Right. But then we don't have any other choice. So once the lockdown ends, I can finally go to lab, and we did the DNA sequencing. So first we ran the amplicon sequencing, and then we did a Sanger sequencing, where we utilized a lot of more genes to actually be sure what the species actually is. Yeah, but the thing is, I can't show you what the species that we got. It is considered confidential for now, because my supervisor is planning to apply for patent application for the whole process, not to patent the name of the microbes, but the whole process. So he would like this kind of microorganism to be confidential for now. But I can say that we got one Bacillus species and of course one Aspergillus species, but it is not the same as the one that we identify with the MALDI, unfortunately. So we can't rely on the MALDI, right? So, of course, we did the same thing, like we have done on Aspergillus and Engeodontium before. So we run on the microbial growth. We want to see how much they are produced, like after 30, 60, and 90 days. And we see that for the fungi isolate, we call this one FunX for now. So we see that the growth increase after 30, 60, and 90 days. And we can see that using UV and heat-treated samples, they grow more rapidly compared to the control. This is the one without any polymer being used as a substrate. Okay. So, but for the bacteria, on the other hand, we can't use biomass to actually measure its growth. Okay, because it is actually very hard to get bacterial biomass, because they are very way tinier than the fungus. So basically, we did a CFU. This is called a colony forming unit, and optical density, where we use UV spectrophotometer to measure the turbidity of the media. So the more turbid the media, the more microbial, the more bacteria presence in the media. So you can see that in both, the growth of the microbes, the growth of the bacteria starts to decline actually after 45 days of incubation. So it is, I can say it is quite contrary to the fungi over here. So because the fungi continue to grow throughout 90 days, but the bacteria on the other hand started to die on me after 60 days. Okay. So so we also did like the same thing. We did weight loss because we've also fed the polypropylene on this new isolate. We did weight loss determination, and we see that fungus, this fungal isolate produces higher weight loss percentage compared to bacterial isolates. And you can see also the trend here, same thing. And we also did a reduction rate. We calculate the reduction rate. Of course, it will be highest on the heat-treated samples, and the half-life will be opposite. So the control will always be there's never any degradation occurs on the controls. Right. So this is just a simplified version. I just want to show on the weight loss only. When we compare between the new isolates and the previous microorganism, the previous fungi, the Aspergillus and Gerontium album, you can see that the new isolate, the fungal isolate, actually produces the highest weight loss compared to the previous fungus. So, and also it shows a much more higher degradation rate, weight loss compared to the bacteria isolate. So, but this comparison is only for the heat-treated film. So, but I just want you to see like the differences at one go. Right. And same thing, we also did like the correlation analysis. We want to see which one is the highest. Like the heat treatment will be the highest. The lowest will be the UV treatment. But for the bacteria isolate, you can see that there are some negative correlation. So the negative correlation here means that the weight loss and the bacterial count does not have a strong relationship, because we see that the bacterial count declines after 45 and 60 days. But the weight loss somehow increased, but only a little bit. So we can see that the correlation is not strong in terms of how the bacteria live and how much the weight loss that we got. Right. And then same thing, we also did the FTIR analysis. And the best thing here, we see that the peaks corresponding is also exhibited an enhancement compared to the control. So that's what we want. There are carbonyl bands present. And also the TGA analysis shows the decline, especially in this one, after from 30 to 90 days of incubation, compared to bacterial isolates. So bacteria is on the right and the fungi is on the left. You can see that the onset temperature where the degradation started is much lower after 90 days for the fungus compared to the bacteria. So we can say that the polymer incubated in the fungal has been affected, has been thermally affected. So the thermal stability started to decline more compared to the bacteria isolates. And we also did this SEM analysis. We can see that how these two shows the differences between the before and the after the incubation. You can see that there are a lot more damage, but you can see a more prominent biomass on the film, on the polymer compared to the bacterial isolates. And we can also see in terms of carbon numbers, the carbon percentage, there are declines in terms of the percentage from the control here and after the incubation with the fungi and also bacteria. Right. And then for the last part of this, we also conducted a GCMS analysis. And for the fungal isolate, this is only for the fungal isolate. We have like these two esters and also the acetic acids, which indicate a much lower carbon number. So we have like C8 carbon number compared to the previous one. So the funky isolate actually produces a much lower carbon number. So it means that they are able to degrade more compared to the previous fungi. So they produce even C8, the acid isopropyl esters. So we can conclude that these fungal are able to degrade more, and when they produces a much lower carbon number, they are able to uptake and they are able to consume and assimilate this polymer as a carbon sources. Right. And then, so this is the last part of my research project. So we did an enzyme assay analysis on these three. So we selected Aspergillus, Angiodontium, and the fungus species only to determine the enzyme assay or lactase enzyme assay activities. So since the bacteria isolate does not show any promising degradation rate, so we decided to just focus on these three types of fungi, right? So this part actually shows the lactase production. If you can see between the left, the Aspergillus and Angiodontium, and the right, the fungal, you can see that the amount of lactase produced after 90 days are actually much higher compared to the Aspergillus and Angiodontium. So the new isolate actually produced about 3.5 nanocatals. That is a unit for the enzyme. But for the previous fungi, the Aspergillus teres actually produced the highest of about 2.4 nanocatals. So the new isolate produced a much higher lactase. So we assume that this lactase enzyme actually contributed more. So that's why the weight loss for the new isolate is much more compared to the previous fungi. Right? And then we also did a correlation analysis to see what are the relationship between the weight loss and also the lactase activities. So you can see the one that are circled, especially for the new isolate, the relationship between weight loss and the lactase activity is the highest compared to Angiodontium album and also Aspergillus teres. So we can conclude here that the lactase produced, the higher the lactase produced, the more weight loss can be caused on the polypropylene. And also we can see that the relationship between the biomass and lactase are also highest in the new isolates and the fungal isolate. It shows that the higher the biomass, the much more lactase activity could be observed, right? Okay, and then this part we actually propose a mechanism. Finally, we propose a mechanism like what happened during the pre-treatments and also what are the products that we got. Like finally, what are the end products that we might be able to use downstream. So these are the proposed mechanisms. So this one, this is not a confidential patent things, but we would like to publish this the mechanism first before disclosing it to the public. So basically, the reaction, I can't show you the detailed reaction, but after the pre-treatment, there are oxidation and free radical formation. So after that, they undergo like a Norwich type 2 and produces hydroperoxides, aldehyde, and ketones. And that's what we got after we analyze using the GCMS, right? But I can show you this. This is a general degradation mechanism that I proposed, and I put it in my thesis. So hopefully the examiners understand this. So basically this illustrates the pre-treatment process, what have been done, what happened to the polymer during the pre-treatment process, and what happened during fungal action. So basically this overall reaction occurs in the presence of oxygen. Okay, we didn't do any abiotic degradation. So it leads to the formation of radicals and hydroperoxides. And the oxidation of the polypropylene chain results in the formation of the carbonyl groups, including aldehydes and ketones, the one that we found during GCMS before. And then the fungi will act on the surface of this polymer by the action of enzymes. So in this case, in this research, I only focuses on lactase enzyme because I found that in the literature that lactase is one of the most prominent enzymes that are responsible for the degradation of polypropylene or any types of polyolefins or synthetic polymer. But there are also actually other types of enzyme. I think I know like more than 10 types of enzyme that are also responsible, that are able to degrade large chain polymeric materials. So among them are like manganese peroxide or lignin peroxidase. So, okay, so basically after the action of enzyme, they are able to produce like a shorter fragments, or we call it oligomers, and it undergoes oxidation, produces carboxylic acid. So we got, for example, the tetradecanoic acid after analysis with the GCMS. So we believe that this kind of short chain polymers are able to be consumed by the fungi through acetyl-CoA, and it will enter Krebs cycle. So this one is assimilated within the fungi, within the fungi cell or mitochondria of the fungi. So, so this compound are further metabolized in this Krebs cycle, ultimately generating carbon dioxide and water as the final degradation products. But for this part, so I put it like a line here. So this is the ideal mechanism end product, but we are unable to actually test on this. Okay, we can actually just assume the end product until the GCMS. I mean the final end product that we got, but whether or not they are perfectly assimilated or mineralized by the fungi has not been tested. But this is the ideal mechanism end product. So basically, if the fungi are able to assimilate or degrade or actually reduce the weight of the polymer, they are able to mineralize the polypropylene. So this is the ideal mechanism that we actually predicted. So, right. So the last part, just a conclusion.
Okay, some conclusions,
Yeah.
okay.
So yeah, this is just a conclusion. I just want to like encapsulate the degradability of the Aspergillus and Geodontium album. And then we found that the new isolate actually produces a better degradation rates compared to the previous fungi. So hopefully someday we could like explore more on these new isolates and actually enhance the degradation of polypropylene or even other types of polymer. And based on the lactase enzyme that we got, hopefully one day we can actually explore other type of enzyme that are produced by this fungus and isolate them and use them to degrade maybe other types of polymers other than polypropylene. So yeah, I think that's the end. Thank you very much.
All right, thank you. So maybe one question, because students have to take off here.
Oh, okay. Did I
Yeah,
take too much? I take too long, sorry.
that's all right. How did you go about selecting those two microorganisms? Was there like prior prior information that you access? Is there a library of enzymes, of microorganisms that degrade polymers? I'm not sure, how did you select the microorganisms?
Okay, so the microorganisms were actually isolated from marine water. So someone from a sea bin project, I'm not sure you've heard of it before. They actually collect garbage from the ocean. So they collect a few types of plastic from Darling Harbour, Sydney. So they bring us the plastic that of course haven't been washed or anything. So I took up the plastic and I sterilized them and I started to grow microorganisms that are actually stick or inside or reside inside of the plastic. So when they grow, so when they grow on the plate, on the media plate, I can see that there are a few types of other colony, or there are different microorganisms actually present there. But the way we select them, we just took up two that are dominant. We can see that there are a lot of similar structure, and then we just select them. So this selection is without prior knowledge of whether they can actually degrade or not. But the best thing is that they are able to grow on a plastic that we get from the marine water. So it means that somehow they can grow on this inert material. So in a way, we assume that they are able to consume the plastic material from the marine water. So we thought that maybe they have the ability also to degrade polypropylene if we do it in vitro in the lab, something like that. Yeah.
All right. Well, thank you very much. I will be staying in touch with you, and hopefully you will hear very soon that you have successfully defended your dissertation. So thanks
Thank you
again.
so much. Thank you, everyone. I hope you get something out of it. I know everyone is tired, so.
Yes,
But if let's say if you have like a further questions or want to ask me anything, just keep in touch. Yeah, you can just give me my email address, or you can give me you can give them, sorry, my email address.
All right, I will.
Contact. Yeah.
Okay.
All right.
Thank you
All
very
right.
much.
Thank you so much, Desmond. It was a pleasure to meet you.
Good, good, good. Nice meeting you as well.
Thank you so much.
Okay, take care.
Yep, you too. Have a good evening or good night.
Yes, and you have a great day.
Yeah. Thank you so much.
Okay then, take care.
Take care. Thank
Bye-bye.
you.
Okay,
Bye.
bye.